Hello, my name is Omar Abasad. It's 2:17 p.m., June 12, 2020. And today I'm going to read an article that I got in my inbox. They've censored Dr. Mercola. So I subscribed to his newsletter and I bought some of his products, which I was happy with. A little bit on the expensive side, but good product. This is an article on the coronavirus. And it says undetectable engineering methods used to create SARS-CoV-2. So the bullet points at the top of the article. So it says, story at a glance, gain of function refers to experiments in which a pathogen is altered to give it new or added functionality, such as the ability to infect humans when before it could not, or increased infectiousness or lethality. SARS-CoV-2 is the only coronavirus with a furin cleavage site. Not even distant relatives of COVID-2 have it, and the coronaviruses that do have it share only 40% of COVID-2's genome. Some experts have pointed out that this novel function couldn't possibly have risen naturally. The fact that SARS-CoV-2 does not have the RBN amino acid sequence predicted by computer modeling does not rule out the possibility that it was genetically engineered or otherwise man-made. US and Chinese researchers have genetically engineered bat coronaviruses using methods such as seamless ligation procedures that leave no sign or signature of human manipulation. Another way you can alter a virus is in a lab without genetic engineering is by culturing the virus in cells that have human ACE2 receptors. Over time, the virus can thereby adapt and gain the ability to bind to that receptor and infect humans. So just to go into the article then, is the COVID-19 pandemic the result of a man-made virus? And if so, what does that say about mankind's ability to safely conduct gain-of-function experiments? Gain of function refers to experiments in which a pathogen is altered to give it a new or added functionality, such as the ability to infect humans when before it could not, or increase infectiousness or lethality, for example. A decade ago, Anthony Fauci defended and promoted gain-of-function research on bird flu viruses, saying such research was worth the risk because it allows scientists to prepare for pandemics. In reality, this kind of research does not appear to have improved government's pandemic responses at all. If anything, it's a curious coincidence that the very viruses undergoing gain-of-function research are the ones causing pandemics. Say that again. It's a curious coincidence that the very viruses undergoing gain-of-function research are the ones causing pandemics. As noted in the 2016 paper, Gain-of-Function Research Ethical Analysis, even if gain-of-function research does lead to improved and effective control measures, laboratory accidents or malevolent action in which souped-up pathogens are released can still result in casualties numbering in millions. So just reading the first line of the paragraph here, it's a quote from Rethinking Biosafety and Research on Potential Pandemic Pathogens, a 2012 paper. If accidentally released mammalian transmissible influenza H5N1 virus viruses could pose a greater threat to public health than possibly any other infectious agents currently under study in laboratories, because such viruses likely combination of transmissibility and virulence to humans. So there was an issue. Such potential pandemic pathogens, as they've been called, jeopardize not only, so I'm feeling to sneeze here. And it happens every single time I'm reading. Such potential pathogens, such potential pandemic pathogens, as they've been called, jeopardize not only laboratory workers and their contacts, but also the wider population who should be involved in assessments of when such risks are acceptable in the service of scientific knowledge and made salty and major public health benefits. The US put a moratorium on government funding of gain of function research into SARS, MERS, and avian flu in 2014. Public tolerance of such risk may be the ultimate determinant of what types of research are allowed to proceed. I'm leaving out a lot of stuff here because it's a very detailed and long article. Again and again, scientists have called for public transparency, saying the public should be part of the decision process, seeing how our health is at stake and it's our taxpayers' money that's being used to conduct this research. So funding pause didn't hold all gain of function research. So it was put on hold, then it was lifted in 2017. That's the research, the funding. 21 gain of function research projects were placed on hold when funding was paused. These studies were initiated before the US government deliberative process research funding pause on selected gain of function research involving influenza, MERS, and SARS. At the end of 2017, the moratorium was lifted and new guidelines put in place. Okay, so, and the review process for gain of function were issued. So skipping quite a bit ahead here. Because of the heavily censored media, any quick online search will easily lead you to believe that there's no evidence of SARS-CoV-2 being an engineered virus, but mounting evidence points directly to that being a reality. So this is the gist of the whole article. With some experts pointing to as a smoking gun evidence for it being a man-made virus at an antiviral research paper. So there's a paper written up. The spike glycoprotein of the new coronavirus 2019 nCoV contains a furin-like cleavage site absent in COVID of the same COV, absent in COV of the same clade, published in April 2020. So according to these papers, again, I'm leaving out a lot of stuff here. SARS-CoV-2 is the only coronavirus with a furin cleavage site. Not even distant relatives of SARS-CoV-2 have it, and the coronaviruses that do have it share only 40% of the SARS-CoV-2 genome. So it's all pointing to it being man-made. Logic behind man-made claims. Any anonymous, possibly Chinese researcher has, I'm sorry, let me do that again. An anonymous, possibly Chinese researcher has also discussed the scientific evidence supporting the claim that SARS-CoV-2 is a man-made virus in a blog called Nerd Has Power. The anonymous blogger, who refers to him or herself as a nobody scientist, points out that there appears to be a concerted effort to promote the idea that SARS-CoV-2 is a natural occurrence. Not nearly as much literature or other forms of substantial writing have been put out to describe or argue for the other possibility that this is a man-made, this virus is man-made. He or she then goes on to explain the importance of the S1 and S2 spikes section. So there are differences in the viruses that can't be explained naturally without going, and he goes on in a lot of detail. So I'm not going to go read it. I'm not going to read all of it. SARS-CoV-2 unlikely to be the result of a recombination, which is probably a natural evolution. And he goes on to discuss that as well in a lot of detail. Therefore, at the very end, the conclusion, therefore, the chance that the furin cleavage site in the Wuhan coronavirus was obtained through recombination with another furin cleavage site containing coronavirus is very low. So I'm going to post a link to this. And I have something cooking on my stove, which you probably could hear boiling away. So I'm not going to make this very long. A lot of people say that when people talk about the virus being man-made, the people who are claiming this possibility are conspiracy theorists. Well, these are doctors who are coming out and pointing out the differences in the virus that occurs naturally as opposed to man-made. So I guess there's a logic behind the way viruses evolve. And the people who are studying them will expect certain mutations to take place and certain things that will take place in the mutations. And they're not finding it in what they're studying. So the conclusion is that it is man-made rather than natural. So conspiracy theorists are not always conspiracy theorists, is my point. So a lot of things that people are claiming that is contrary to what's being put out in the mainstream media, contrary to what the government is telling us, contrary to what other doctors are telling us, is sometimes the truth. In other words, we are being bamboozled. And since I have to go see my pot, look after my pot, I'm gonna just end this here and pick it up another time.